This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 98, recorded on February 5th, 2014. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello, everybody.
How are you doing?
I'm doing fine.
Just fine?
Oh, no, I'm doing well,
You sound so far away. Well, I guess you're a few thousand miles away.
That's it.
Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello.
How you been?
I've been staying warm.
You've been in Ann Arbor. You haven't gone anywhere exotic?
Not that I can remember.
Do you have a lot
teaching.
of...
I'm teaching now, so I'm sticking close to home.
What are you teaching?
I am teaching a first-year seminar for freshmen who, of course, haven't declared majors, and it's called Current Topics in Microbiology.
Wow.
So we're just picking highlights and talking about, West Nile virus and the measles outbreak. And,
you do some viruses. That's good.
Yeah. Oh, yeah.
Very good.
Vaccines.
Good to hear it. Yeah, the vaccine is becoming a political issue this year. Isn't that ridiculous? It's just nuts. Our governor, the governor of my state, says people should have a choice. And I'm thinking, now let me get this right. When you're drafted, you have no choice and you have a much higher likelihood of dying. So what's the logic here?
And seatbelts.
Seatbelts is another one.
And
Oh, you know what? One of the let me introduce Michael, and then I can give you this one from Charleston, South Carolina. Michael Schmidt.
Hello, everyone.
How you doing, Michael?
I'm doing well, thank you.
And you don't have snow, Michele? Do you have snow on the ground?
A good foot.
Wow. I heard one of the Carolina senators has recently said, and you guys might know this, that employees of restaurants should not be required to wash their hands.
No.
As long as they put a sign on the window. I saw that.
Say it wasn't South Carolina. Please say it wasn't one of my two senators. Please.
Who was one of the two Carolinas. I just heard it at a faculty meeting. His proposal is that you put a sign up saying employees in this restaurant do not wash their hands, and that'll take care of everything because people won't go to that restaurant.
Transparency.
Here we figured out how many 100 years ago that you had to wash your hands to avoid transmission of disease. And now we're going back. I don't understand this. Since when does politics get stupid? Oh, I'm sorry.
Especially the public schools. We're obligated to keep children safe when they go to school. And if you choose not to vaccinate, that's fine. You can choose not to have your child at school.
It was Senator Tom Tillis, a Republican of North Carolina, said at a bipartisan policy center meeting that maybe the government should not require food workers to wash their hands after using the bathroom.
Any reason why?
Oh, it's an infringement of personal liberties.
Yeah.
Tillis said he was at Starbucks in 2010 talking to a woman about regulations and where businesses should be allowed to opt out. His coffee companion challenged him, asking whether employees there should be required to wash their hands.
Why should they do? How about food inspection? Isn't that an intrusion in personal liberties?
Yes.
It's your right to transmit infectious diseases to other people.
Right.
Absolutely.
But I think he's getting quoted out of context. He says, as a matter of fact, I think this is one where I think I can illustrate the point, he recalled telling her. I don't have any problem with Starbucks if they choose to opt out of this policy, as long as they post a sign that says we don't require our employees to wash their hands after leaving the restroom. The market will take care of it. Bad. So he's a free marketeer. It's one example.
What do you mean free marketeer? He's making Starbucks put up a sign.
Yeah,
Come on. That's government interference.
That is true.
He's
anti-regulation, but he wants signs.
Oh, well. Okay. That's a little interesting stuff. Then, Michael, why don't you send me a link to that? So I'll put it or stick it in the show notes.
Okay.
Put a link to that.
It's from the Washington Post.
Okay, today we have a snippet and a paper. And let's start with this snippet from Alio.
Okay, this is a snippet about bacteria snipping. About how to divide. The article, which appeared in Nature Communications, is called Size Independent Symmetric Division in Extraordinarily Long Cells. And the authors are Pende, Leisch, Gruber, Wodicka, Heindel, Ott, Den Blauwen, and Bulgarese. And this is a group of people from a couple of places, Vienna, Amsterdam, and Bremen, Germany. And there are two things about this paper which are quite remarkable. One of them, let me just give you the punchline first. We're dealing here with bacteria which cover marine nematodes. These are guys who are 1 to 2 millimeter long, and if you look at them under a scanning microscope, they are covered with bacteria. There are two kinds. Actually, I'm digressing, but I'm doing it on purpose. There are two kinds of bacteria, or two kinds of nematodes covered. They're completely covered by the bacteria, by the way. In one of them, the bacteria are crescent-shaped, and the two ends of the crescent are stuck on the surface, on the skin of the nematode. And what it looks like, and you have to see this to believe it. It's unbelievable. Completely covered, head to toe, and it looks like a braided rope. It's the best I can describe it.
It's absolutely beautiful.
Yeah, it is. It's beautiful.
And more, as remarkable are the other ones where the bacteria are stuck by one end only, and so they're sticking out, and these guys look like woolly bears. I mean, they're so cuddly. All you want to do is cuddle to them. You got to see this to believe it. Anyhow, so there's two kinds of bacteria, two kinds of worms, and they have a characteristic in common, namely that they're quite diverse in their length. They go in length from something like a few microns to 140 microns. The two vary a little bit. The range of one is 16 to 140, the other is 45 to 3 to 45. Anyhow, the point here is that if you look at where they divide, where in the cell do they divide? They all divide in the middle. Whether they're long, whether they're short, whether they're stuck on both ends or stuck on one end, they all divide quite precisely in the middle. And these people went on to study this in some detail. Obviously, you can't cultivate these guys, by the way. You have to look at what you get. That's it. And so they looked at the location of FTSZ, which is a bacterial tubulin, which is many of our listeners know, makes a ring at the site of division, a constricting ring at the site of division. And by and large, the site of the FTSZ ring is the site of division, and this is in the middle, quite precisely. By the way, the ring does not appear on all cells because younger cells do not make the ring. They only make the ring close to division time. Anyhow, that's the story. So let me go back a little bit about the bacteria. These are gamma proteobacteria, which are sulfur oxidizing. That is, they take H2S, and I guess these guys take oxygen as an electron acceptor. Others take nitrate, and they produce energy this way. They are autotrophic bacteria. And do they feed the nematode with the products of their metabolism? Well, nobody knows, but they might. There are other worms which depend entirely on the metabolism of bacteria. So anyhow, so the point here is long and short, they all divide in the middle. So division is not due to the cells having reached a certain length. Takes care of that theory, at least for these bacteria. People think about division in E. coli as being due to one or two things. Either a ruler, the bacteria have a ruler to measure their size, and when they reach a certain size, they can figure out the middle. And the other is that it's a timing thing. And this is being argued quite vehemently. And here, at least, you can say it is not length. That's about all you can say. So
Elio, when I was looking at your little snippet, I remembered reading once upon a time an article that Andrew Wright put out, literally with the title, "How Does a Bacterium find its middle. And so it's up in Nature Structural Biology from 2000. So I'll put a link to that in the show notes for everyone.
Well, I'll tell you, a bunch of physicists have been writing a whole bunch of papers about this, and they are not in agreement about what it is that sets off division. But there are two issues here: the timing, when does it happen, and the position. The position is what you're dealing with here. There is a lot of talk about the MIN system, which is a bunch of several oscillating proteins, which go back and forth, in some cases from one pole to another. In some cases, they sit at the poles, and that they impose on the bacterium a knowledge of where the middle is. And then there is something called nucleoid occlusion, which says that wherever there's a nucleoid, there cannot be division, because if there were, the septum would guillotine the DNA.
Yeah.
Which is not good for business. Anyhow,
it's not good for business.
It's not good for business. It does not quite explain the quite remarkable precision of the site of division, being really quite in the middle of the cells. But anyhow, this is adding, as I say, it's adding an interesting fact, an interesting study to this big question. But in addition, it is so much fun to look at these nematodes. And let me tell you that one of these authors, Silva Bulgarese, wrote a paper, I'll put that in the notes, in which a different nematode has bacteria on it, but instead of lying flat, more or less, which is what the guys with the two-pole sticking do, these guys are perpendicular. And what's more, they divide perpendicularly. They don't divide in the middle, they divide lengthwise. We had a piece on this, and we called it Bacteria Learn the Long Division.
It's sort of like a hot dog bun.
Like a hot dog bun, exactly. And that is so incredible. Why would they do that? So the world of nematodes is a bacterial garden of delights.
So, Elio, this figure, the one that looks like a rope, right? Figure 1.
Yeah.
Those. What are we looking at? Are those the nematodes there, all entwined?
Yeah. No. You're looking at the bacteria.
These are the
bacteria surface of a nematode.
And on the bottom is the actual nematode. Is that correct?
Yeah, in different magnification. I mean, this is
okay.
a totally different magnification. So it's so unbelievable to look at. And as you say, you don't know what you're looking at because this is unheard of in biology. You think it looks like a rope? Braided rope.
For sure. It has a little periodicity too. It looks like
that's right.
Beautiful.
That's right. It has
it looks
periodicity.
Braided.
Yeah, and these are very long bacteria, right? They're filamentous.
That's right. And in the other case, there is not a good picture in this paper, or there is in the supplemental material. So people who look at this paper, go to the supplemental material, and you see the woolliest worm you've ever seen. It's unbelievable.
The woolliest worm. I like that.
The woolliest worm in creation. It looks like it's just cute. I say it's cuddly.
Beautiful. That's the true nature of a snippet, something that makes you go, wow. Thank you, Alio. That's pretty neat.
My pleasure.
Our paper this week comes to us, was sent by Patrick, who is, I believe I think he's a student out at the Fred Hutch Research Center in Seattle. I've met him a few times. He writes, I thought you and the rest of the TWiM folks would be interested in the following paper. Transferred interbacterial antagonism genes augment eukaryotic innate immune function. Matt Doherty, the Malik lab, who you know, co-authored the paper with Sime Chu from the Mugu lab. We've had a lot of fun following this story in the lab. I think it's a beautiful example of interdisciplinary approaches coming together to uncover a very cool bit of biology. Happy holidays to the entire Twifecta. So, of course, he sent this at the end of last year. So we're just getting around to doing it. So I am looking for a paper this week, and I said, this looks like a good one. I read a few lines, and it was quite clear that this is a pretty cool paper. So it is published in Nature. The authors are Chu, Doherty, Peterson, B-Boy, Yang, Jutras, Fritz Leyland, Farron, Harding, Jacobs, Wagner, Yang, Vollmer, Malik, and Mugu. And they are from the Department of Microbiology at the University of Washington in Seattle, and also Fred Hutchinson Cancer Research Center. There are also some people from Newcastle University over in the UK, Indianapolis, Indiana University School of Medicine, Yale University, UCSF, and Yale again. Yeah. So this is truly an interdisciplinary effort. Wow. It's amazing. A lot of authors.
It's a dream
there.
team, isn't it?
Amazing.
5 labs with different expertise.
That's the way it's going. Anyway, this is all about what we call, and have mentioned before on TWiM, horizontal gene transfer. Now, we all know that organisms can acquire new functions by the easy route. They can just get a gene.
By the way, can I tell you a funny?
Yes, of course.
In an undergraduate class, I once asked a question about horizontal gene transfer, and one student says, that's the transmission of sexually transmitted diseases.
Very nice.
And the answer is correct.
Anyhow.
So it's a good way to get new genes and new functions, and it happens. And there are lots of bacteria out there, so they have a lot of DNA to contribute, and they have contributed genes to microbial eukaryotes like algae and other protozoa. But metazoans getting genes from bacteria is a little bit harder to pin down. We've actually talked about a couple of stories on TWiM, one where pieces of bacterial DNA were found inserted into eukaryotic genomes, human genomes. We don't know what to make of that. We don't know if they're expressed, if they have any function. There are a few examples, though, of transferred genes from bacteria that confer beneficial traits, and we talked about one on TWiM 89, how a gene from bacteria protects a tick from plant cyanide poisoning. All right, that was a really cool story.
I believe another example are some viral genes that during viral pathogenesis cause cell fusion or syncytial formation.
Oh, yeah.
And my understanding is that placenta formation by a number of mammals has co-opted those viral genes.
That's right.
So the endogenous retrovirus, right?
That's right. So the fusion glycoprotein of the retroviral, the gene encoding that, is in the mammalian genome, and it is called syncytian. And it makes the syncytial trophoblasts, which are a layer of the placenta. They're all fused, and if without that protein, they wouldn't exist. So, mammals that give birth, live birth, owe it to a virus, apparently. And this has happened multiple times.
Independently.
Yeah.
Not the birth, but the transfer of the...
Yes, right. The transfer.
I bet you makes you very happy, Vincent.
Well, yeah, I like when viruses are doing good things. I collect examples and I tell people all about them. Anyway, any gene that's transferred to another organism should give it a new function. And so one of them's they were studying in this paper bacterial genes called TAE, type 6 secretion amides effectors, and these are enzymes that are injected into your enemy bacteria that degrade the bacterial cell wall. There are 4 families. They're called TAE 1 through 4. They're different sequences, and they have different specificities. They cleave peptidoglycan, that component of the cell wall, and members of the 4 families are different. These authors were studying the evolution of these genes, and they say by accident we found that there were related sequences in eukaryotic genomes. So now you can just go into the database of sequences and look at genomes of all sorts of eukaryotes, and you can even get some RNA data. In other words, what mRNAs are expressed. And they found these TAE genes and quite a wide range of eukaryotic metazoans like amoeba, crustaceans, ticks, mites.
You don't mean metazoans.
What did I say?
You said metazoans. You mean eukaryotes.
Multicellular metazoans. Well, eukaryotes, I mean. Yeah. What else? Various things that live in the ocean, sea anemones and lancelets, these odd-looking long fish, acorn worms. So they find that you can find the sequences in a lot of places, okay? They don't appear to be contaminants, because that often happens when you sequence a genome. Sometimes there are bacterial contaminants in the genes show up and you wonder if they're part of the genome or not. But these related genes have introns, intervening sequences, which are not present in bacteria. And they have genes on either side of them, wherever they're finding them. So, you would not expect to see that all the time. If they're just contaminants, they would just be randomly isolated. So they appear to be rare. So they call them domesticated amides effector genes, or DAE. I like this use of the word domesticated, right? We've grabbed them from bacteria and we put them to work like domesticating an animal, right? 3 of the 4, so you can look at the sequences and do bioinformatics. You can see that 3 out of 4 of these 4 TAE families have been transferred from bacteria at least 6 different times. All right, so that's just based on where we are in sequencing genomes of various organisms. There could be more because we haven't sequenced everything on the planet.
And the database is only as good as the databases.
Yeah. One of them in particular, DAE2, is found in 10 different species of ticks and mites. And they calculate that this happened by a single horizontal gene transfer of the bacterial TADE2 gene to a common ancestor of ticks and mites about 400 million years ago. So you can figure this out based on the phylogeny of these species, when the common ancestor existed, when ticks and mites separated, and so forth. What's also interesting is that at least one mite genome lacks this gene. So in addition to gaining them, they've also lost them, which is interesting. In arthropods, this DAE2 gene probably went into that lineage 550 million years ago. There are 2 scorpion species and a horseshoe crab that have these genes, and 400 million years ago in mollusks and an annelid.
Do you think that these genes came about because the animal swallowed the bacterium, or it became an endosymbiont, or an infection, an infective agent, or any of the above?
Yeah.
All of the above, I'd vote.
Yeah, I would bet that all of those, and maybe more, sure. Right. Who knows? It just it probably happened randomly, and then some use was made of the gene in evolution, right? It provided some advantage, and it remained.
Right.
Other eukaryotes, choanoflagellates, sea anemones, acorn worms, lanceolates. DAE4 was acquired over 800 million years ago, before the base of the metazoan lineage. So they're there, and they appear real. But the real question is, of course, are the genes functional? And without doing any experiments, they can just look at the data and get suggestions that they are. First, there's evidence of what we call purifying selection. So you can look at the sequences of these genes throughout phylogeny, and you can see that certain alleles, certain sequences go away. And that suggests that they're deleterious in some way, so we call that purifying selection. In addition, these genes have acquired the eukaryotic signals for secretion out of cells. So that implies that they've been functional. And finally, these are enzymes that have catalytic centers that carry out the enzymatic activity. Those are still there. They're conserved, so it says, looks like the enzymatic activity has been maintained.
Well, that was one of the fascinating pieces about how it could swap in a eukaryotic signal sequence. I mean, how does it do the selection for it? It must be a tremendously important aspect of conferring fitness to the eukaryote in order to drive the selection for that. It's got to be a mutational event. Or do you think it was just by happenstance that it...
I think it would happen that it recombined in some way to pick up the secretion signal, and, may have happened in multiple times, but there was some pressure that kept it there as a secreted protein.
And secretion signals aren't all that specific, not like the catalytic center of an enzyme. Yeah. So it might be easier to acquire by random mutation.
Still, it's pretty remarkable it all came together that way. And you wonder really about what the selective pressure was. And
I can't wait to get to the end of the story, because there's a beautiful experiment that gets at that.
I want to have a lens to look at these. I wish I could look back at exactly the moment where the selection happened and see it, right? We can't do that, but I wish we could do that. Finally, more bioinformatic evidence. You can find RNA. So these genes are transcribed into RNA of these genes. You can find that in ticks, you can find it in amoeba, and you can find it in this lancelet. So again, you can just look in the database of transcribed genes of RNAs and find that information. So that's all the bioinformatic information. You just need a computer to do that. Now you have to do some experiments, right? Because you have a hypothesis. And what they do is the first thing, they take the genes encoding these DAE proteins. They take them from ticks, from amoeba, and from lancelet, and they put them into bacteria. They put them and produce the proteins. They purify the proteins. And they ask, can these enzymes hydrolyze peptidoglycan? And they use peptidoglycan from E. coli. And if you remember, the peptidoglycan is a polymer of sugars linked to amino acid chains that makes kind of a mesh in the cell wall. And the DAE proteins cut at different places among the amino acids that are branching off from the carbohydrate. So day 2, they find that day 2, day 3, and day 4, DAE, not DAY, by the way, shows substrate specificity that matches the homologs of TAE in bacteria. All right, so the substrate specificity has been maintained. And they can do this by cleaving the peptidoglycan. They do mass spectrometry, or actually HPLC, and they can know exactly how these are cleaved. Really cool technology. So this result supports the idea that the homologs have been retained because of their enzymatic activity, because they are still enzymatically active. What are the proteins doing in the eukaryotes? So we know they're enzymatically active. They can cleave peptidoglycan. What do they do? So they looked at the deer tick, which is Ixodes scapularis, and it's, of course, the vector for Lyme disease and anaplasmosis. So this is a medically important tick. They find by looking for the protein, by dissecting the animals and doing Western blot, they find the day 2 protein in the salivary gland and in the midgut. So these ticks, remember, they take blood meals. Of course, that's how they transmit bacterial infections. So the salivary gland in the midgut is going to interface with bacteria. So they're starting to think maybe day 2 is involved with bacterial defense.
No, defense against bacteria.
Or that. Yes. I wrote it here, bacterial defense, but defense against bacteria. Does Borrelia make ticks sick? Do we know?
Typically not. It doesn't impact on, I don't think, the total population of ticks, but it makes their host sick.
Yeah.
And if the tick is going to want a healthy host to reside on, and the host goes down because of an infection from Borrelia, or the blood gets too warm from the fever that it causes, because remember, some of the Borrelia cause relapsing like fever syndromes, and so you may be going up and down, and they may not like the warmer blood or colder blood.
You know, I think that's way too
complicated,
anthropomorphized. All right, it's way too human. It's a human interpretation. I mean, this is what I think is one of the issues with this study. I don't know why the ticks would defend against a bacteria that doesn't really affect it. The host business, I don't buy, because you can always find another host. But anyway, I'm not sure why these things are there. Anyway, they look at the ability of day 2 in ticks to cleave proteoglycan of bacteria that it would normally encounter in the environment. So they find that they look at 2 types of firmicutes. They look at the bacilli class of firmicutes. It will certainly degrade peptidoglycan from those, but not Streptococcus pneumoniae. It also degrades the peptidoglycan of Borrelia burgdorferi, and Willy Burgdorfer we talked about. He just passed away not too long ago. We mentioned him on TWiM. He discovered this bacterium. So, it makes sense that some of these bacteria that the ticks would encounter, the peptidoglycan would be cleaved. Now, if you just take the protein day 2 and mix it with E. coli, it doesn't do anything to them. It's only lytic for E. coli when you permeabilize the outer membrane, or if you put a signal on the protein, the day 2 protein, that lets it get into the periplasm. Okay, so it alone is not enough to harm the bacterial cells. So it's got to be together with something else. And they say, well, maybe an antimicrobial peptide, for example, that could permeabilize the outer membrane and let the enzyme in. It would be necessary for it to be antimicrobial. And finally, the experiment that Michele was referring to, does day 2 regulate Borrelia populations in ticks? So they do a cool experiment where they treat ticks with short interfering RNA to knock down the levels of day 2 mRNA. So that will reduce the amount of day 2 protein. They let these ticks feed on mice that have been infected with Borrelia. And then they ask, how much Borrelia do you find in the ticks? So if you check the ticks just after what they call engorgement, they've drunk as much blood as they can take and they're falling off. You check that. There's no difference in the number of bacteria between ticks with or with knockdown day 2. But if you wait 2 weeks later, there's a significant difference. There's much fewer Borrelia in the ticks, which still have day 2. If you take day 2 away, there's more Borrelia. And interestingly, there's no gross change in the overall bacterial load in the tick. So it's somehow something specific for Borrelia, although they say they might not be picking up other minute changes in the populations that could affect that. So here we have this gene which came from bacteria. It's in ticks for sure. It seems to be active. It cleaves the right substrate, peptidoglycan, and its presence is needed to reduce the amount of Borrelia in ticks. And they didn't ask the ticks how they felt with fewer Borrelia, right? I'd like to know. They didn't measure the fever of the tick or any other parameter. So I don't know why a tick would care to have less Borrelia in it, but that's something we have to figure out.
So I have to interject here. I had a chance to have a chat with the first author on this paper, a postdoc, Sime Chow, and she told me a little more detail about that final experiment. So you mentioned that they used RNA silencing technology to reduce the amount of day 2 function. I just want to point out to our listeners that involved microinjecting the siRNA into the anus of these tick larvae.
Ouch.
And she's estimating
Wow.
that some 10,000 ticks sacrificed their dignity for this experiment. And you can imagine that that's technically very challenging. Sime actually traveled to Indiana to Frank Yang's lab, and he's an expert in the Borrelia tick model. So 3 separate occasions she went there for a week at a time to learn this technique, came back and then trained another colleague, Brooke Peterson, who was a huge help in this effort. And she described that it took maybe 6 months to come up with the hypothesis and the model. And that was thanks to a lot of work that she did with Matt Doherty, who it turns out she and Matt knew each other as graduate students in the Bay Area. And then they both happened to be doing their postdoc in Seattle. So it was kind of an informal conversation that got that started. But after a 6-month period, they had this fabulous hypothesis. But then it took a year and a half of this really painstaking work with these tiny ticks and the microinjection to really test their hypothesis and come up with the phenotypic data that showed the gene does confer a fitness advantage to the new host. So just to give you a little insight to what actually goes on in the lab.
That's cool. Yeah.
Yeah.
Ticks are tough to work with. Yeah.
But I think the most remarkable aspect of this story is we know from the last TWiM's discussion of how quickly bacteria adapt to antimicrobials. And the fact that the microbes have not adapted to this day protein.
Yes, I was going to ask you that. Why not?
I don't know. It must confer fitness in a manner that has nothing to do with peptidoglycan. Because you would think that if it was limiting the ability of the population to make cell wall, they would have adapted their cell wall mechanism similar to the way microbes adapt when we give them beta-lactam antibiotics. Because what it's effectively doing is what beta-lactams do. Beta-lactams prevent cross-linking, and what this does is it breaks the cross-link. So maybe it's the chicken and the egg because it's doing it after the cross-link is formed, that resistance hasn't emerged and there's no way to detect for it. But I think natural selection would have altered the peptide between the 2 strands of peptidoglycan to change it. Yeah. So I think the real crux of this paper is why over the millennia has resistance not emerged? Because if we can figure that out, we'll know how to design better antimicrobials.
Yeah, that's a good question, I think. The other question I have, and I submit to all of you, why don't humans have these genes? They might be useful, right?
Although we all know that our normal flora, biota in our gut, are very beneficial too. So you'd need some specificity.
You could. I see. So you don't want to kill off the good ones, but
right.
And you would need to have them in places like the gut and the lung and the skin and so forth to protect you. Yeah.
Mm-hmm. We may have specific ones that will target specific bad actors, but we just haven't asked the question because we don't know what those antimicrobials are. Remember, our selection of antimicrobials is pretty limited. There's about 5 to 6 classes. There's the ones that affect cell wall, the ones that affect protein synthesis, the ones that affect RNA and DNA synthesis, and then the instructive antibiotics that interfere with certain aspects of metabolism. I'll bet if we hunt looking for some unique aspects of metabolism that humans have to inhibit bacteria, the example of one of these instructive antibiotics, of course, is an old one, sulfa, that we may be able to hunt. So that's where, if you're going to do a bioinformatic exercise, ask the question about some of these other targets for which, we've discovered antibiotics. They may be actually in our DNA.
The thing that comes up here is that we are not affected by a lot of organisms, a lot of bacteria and viruses that affect our cousins.
Yes.
And so we can, there's a huge number of diseases of veterinary medical importance where the organism does not affect humans. Not everything is shared by everybody. And those would be candidates for organisms to which we have a defense mechanism. Obviously, we do. And so that's where one could look.
Mm-hmm.
So let me postulate this. Do you think as society has become more technologically advanced and we've begun to lose our Helicobacter out of our gastric system, do you think that humans, as society became more technologically advanced, acquired one of these genes that no longer allow us to carry Helicobacter in our guts?
That would have to be a pretty recent happening, right?
Right. I mean, the Swedes were the ones who originally described this, and they have this great curve about Helicobacter, but that could explain a lot about why we're losing Helicobacter if it's acquired. Humans have acquired one of these genes horizontally.
But Michael, if this happened, let's say an HGT from a bacteria did this transfer as you're suggesting, it would probably initially happen in just a few people or one person, and to have that one person propagate to the world's population would take thousands of years.
True.
Unless it all happened in thousands of people or millions of people at once, which I think is really unlikely, but I don't know.
I was really impressed with how many independent times this had happened to so many different forms of life, from crustaceans, insects, mammals. And then Sime in our conversation again pointed out that in the same week that their paper was published, Seth Bordenstein's group from Vanderbilt, in collaboration with a colleague at Portland State, also documented a gene transfer, horizontal gene transfer, also an enzyme that attacks bacterial cell walls. But in this case, it was archaea that had acquired the antibacterial. So I think if we step back, this demonstrates how microbes are everywhere and they're providing a selective pressure for antimicrobial mechanisms across all the domains of life.
In this case, I'm not clear on why. I agree it's been transferred. The Tay genes have been transferred many times, so they must be important, but I don't know why. And I think this paper doesn't tell us why they make some suggestions, but I don't think they tell us why the transfer is important. And I think that needs to be sorted out. I'd also like to, I hope that here on TWiM we get to do the paper where they find such a gene in people, in humans, and figure out, because there are sequences in there, but they don't seem to be doing anything. So
another neat thing about this story is that it brought together people with very different expertise. So 5 different labs. And they really were just their excitement about the potential for this new biology motivated them all to kind of step out of their comfort zone and really pursue this very cool biology. And Sime really credits the inspiration that she got from her graduate mentor, the late Tom Albert. He's a crystallographer, a biochemist that had a wonderful career at UC Berkeley. And he unfortunately passed away just a year ago. And Sime was kind of wishing that he had been around to see this work come together, because she says it's really a credit to him and the kind of science that he taught his students.
Nice. That's good.
Yeah.
Well, I'm sure other genes have been transferred and they just remain to be studied, right? It should be interesting to see what they do and what functions they provide. It's pretty neat. So another paper, that other paper, lysozyme, do they know what the lysozyme is doing in the archaea? I would guess not.
Well, they have evidence that its expression is increased when you culture the archaea with bacteria.
I see.
So you can deduce that it would be a defense mechanism.
Yep. That's cool. Nice. Well, this is obviously an area ripe, so we're getting multiple publications. So we'll keep an eye on that. Anything else? Anyone else want to comment any more on this? This is a neat paper. Thank you, Patrick, for sending that. And let's do some email. The first one's from Dave. He writes, "Dear Mr. President, President alum, and President to be." I don't know who the alum and to be would be, unless you guys.
The alum is Elio.
Okay.
The president is you.
And who's to be? Maybe Michele.
Michele.
I don't have any plans.
Well, you just get nominated. And then you say you're happy to serve.
Who said if nominated I will not run? If I'm elected I will not serve? Someone said that. Anyway, thank you, Dave. Alio said Cretaceous, referring to 400 million years ago, and Michael agreed. I thought, wait, that's when Jurassic Park was set. Cretaceous, i.e. in the last era of the dinosaurs, exclusive of birds, from 100 to some 66 million years ago. You guys were talking about how ancient must be the tenuous association of the tiny cyanobacterium with the tiny algal bit in getting fixed nitrogen into the eukaryote. 400 million years ago was just at the end of the Devonian and beginning of the Carboniferous. I had to look that up.
Oh, boy.
Perhaps you meant the era starting with a C, which was not the Cretaceous. Looking forward to contemplating my metabolobiome under the influence of my microbiome and my virome, enjoying my insomniac listening again. Wow. I remain faithfully yours. I guess we like insomniacs who listen to us, right?
Yes, absolutely.
I mean, we don't wish them to be insomniacs, but we appreciate it.
Are you saying we're putting everybody to sleep?
No, you can't sleep. You can't sleep. So I hope we're not.
Well, thank you, David. Anyhow, if I was wrong, I readily admit it. I was not in my comfort zone when I talk about.
Often we're not, and we're brave. I'm glad you guys are willing to do this, because I certainly am.
And that's the whole bit about science is you may not know the fact, but then your colleagues are to set you straight. And we thank you for doing that. And when Elio speaks, he speaks with such authority. So I always agree.
It's okay.
That's what my wife says. And she says I'm usually wrong.
One of my favorite lines is, "I can't be good at everything."
I know you say that quite often, but we don't want to be good at everything. We like to be good. I was talking to someone yesterday, and I said, I think I'm good at one thing, and that's viruses, and not even all viruses. Nothing else, forget. I can't fix my car, etc. Robin writes, for those unacquainted with the armed forces, about the email discussion on food safety. He sends a link to a Navy publication, which is very long. He said, page 65, section 3-501.19, parentheses B. You can find out how long you can keep the turkey out before you shouldn't eat it anymore. The Navy, as you would imagine, has directives on this, and they're all laid out. It's really funny.
But it's very practical. I mean, you don't want a sailor sick on a submarine.
No, you have to have rules, right? When you have a lot of people like that, you have to have rules. Otherwise, things get out of control.
And vaccinations for all.
All have to be vaccinated
before they get on that ship.
You know what, Michele?
Yes.
They don't have any choice.
I'm sure they don't.
You have to get your vaccine.
Of course.
Even against anthrax.
But I guess, no, I'm not going to go there. All right. Thank you, Robin. Nick writes, hello all. Thank you for the wonderful and insightful podcast. I am a somewhat regular listener as I work in viticulture and spend my spring, summer, and fall scouting vineyards, advising grape growers on integrated pest management in Northern California. It was brought to my attention by my girlfriend and dentist that newborn children, is your girlfriend your dentist or two separate?
Yeah.
That newborn children are born with sterile mouths. I am curious as to why this is the case. Could it be that some newborns have colonies of bacteria that are passed on from the mother? Thanks for your thoughts on this question. Happy New Year. All right. Who knows the answer to this?
I can't believe that that's true.
I don't believe it's
true. If it depends whether during the birth, passing through the birth canal, do babies keep their mouth closed?
Well, usually it's full of
I
mucus,
don't remember.
Right? They put, because the delivering physician puts the finger in and clears out the mucus right before they take the first breath.
That's
so
right. Yeah.
I would guess it's not sterile because the mucus is probably coming as it's being delivered, right? Not just from in utero. But what do I know? I can't be good at everything, right, Michele?
and this is
right.
this is an emerging area of research, and as you can well understand, it's very hard to get a mother to do informed consent for you to go and swab her brand new newborn baby, which she doesn't want to give up to anyone. So,
well, and as soon as the baby starts nursing, they're going to acquire,
of course,
healthy, good microbes.
When I introduce this to the dental students, I point out to them the first gift they receive from their mother is as they transited the birth canal, they began to pick up her flora,
right?
And then when they began to nurse, they of course acquired bifidobacterium, which is one of the first gifts, along with the other lactobacilli that then helped facilitated the digestion of milk.
So, Nick, I think your girlfriend / dentist is probably not right about the sterile mouth because, as you've heard. Anyway, Nick also sent us a link to the placental microbiome paper, which came out some time ago.
And I'd just like to point out that ASM on their press site just announced a paper that birth method, gestation duration may alter infants' gut microbiota and influence later health. And this was a study that was from the study co-authors were Yolo and Lin School of Medicine in Singapore, amongst others. And it's a rather fascinating study that was published in MBio. So it's a free paper if you wish to look at it. And it just came out in MBio, and it's an absolutely fascinating work.
Okay. The next letter is from Bethany, who writes, greetings. I have just finished episode 77, so I am slowly catching up. I wanted to say that while I am a layperson, as an elementary school teacher and hobbyist fermenter, I enjoy learning about microbiology and find the topics fascinating on the show. One of the anthropological aspects of food that I find fascinating is chicha or chica.
Chicha. It's fermented maize, corn in the Andes.
And other foods where saliva has started fermentation, although I would hesitate to try this myself.
Well, the way it used to be done is that the ladies in the kitchen would chomp away and spit out the masticated corn with amylase and all. And this, of course, broke down the starch into fermentable sugars. And this is a drink which is used in the Andes, in Ecuador, Peru, Bolivia.
It's also affectionately referred to as corn beer.
That's right.
So any of our listeners in Wisconsin who are into everything corn, let us know, or Illinois.
And Bethany continues, I wonder how much this kind of thing impacts the diversity or monotony of the indigenous population's salivary flora. Overall, I just wanted to say thanks for a good show, and I look forward to getting caught up soon. How would this impact the salivary flora? I don't know.
Generally, these things don't have all that much alcohol. Like the name beer implies, they're not fortified beers, so it's probably nowhere but 1 to
You think
at most 5% alcohol.
You're talking about drinking it, but maybe I think she means is if you chew this, to get it started, does that impact the flora in your mouth?
Well, the sugars certainly could. I mean, depending on what's in the maize,
I suppose.
I mean, they could. You're always selecting organisms by what virtue of what you feed them.
Yeah.
Or the women who had the most optimal flora in their mouth were famous and highly sought after, and so they ended up marrying well and having lots of children. And so,
so that's a good point. So
you accuse me of anthropomorphizing. Michele wins the prize today.
For creativity.
Yes.
So here's the question: if, let's say, there are certain women who are employed to chew the maize and produce it, would having their oral flora influence the oral flora of other people who drink the beverage?
It's not pasteurized, and there are probably microbes in it, but as you reach the alcohol level, it may kill some of them off.
And then other things grow up, right?
Okay. This polling question from Caroline had been sent to TWiM, but I thought we should bring it up here because I know you guys would probably have a better answer. And she asked, are virus infections more likely to be species-specific than bacterial infections? It seemed to me they would be, since viruses rely so much on the specialized relationship with cells for, quote, survival, unquote. So we talked about the virus half, which is fairly species-specific. There are certainly viruses that infect more than one species, but on the whole, they are limited. What about bacteria? Are they...
Well, they range tremendously from Pseudomonas that infects almost everything there is to very specialized bacteria like the gonococcus, which is only known to affect human beings.
Yeah.
So the range is huge.
Especially because the bacteria can grow extracellularly. So I would think there'd be fewer constraints on the bacteria.
Exactly.
But there are some.
Sure.
There are definitely some. And the other thing to consider is that the genus species, in the traditional sense, while you have the same name, it's really looking for a receptor to attach to, because the microbes, when they make an infection, they're not often planktonic. They're actually attaching to us and making a local abscess or doing something specific.
Let me tell you the experience I had. I taught medical students as well as vet students. And I had to learn a bit about veterinary microbiology because I knew even less about that than about human. And what surprised me is that a lot of the major diseases were very different. There are some diseases, there are some bugs that affect both humans and cows and horses and dogs, but there are many which are specific. For instance, Corynebacteria turned out to be a major veterinary organism of veterinary importance, and it's limited in what it does to humans. So there's many examples of that. So if you just go by that landscape, it's surprising how specific bacterial diseases may be. I may be wrong because this is impressionistic. This is not a study I made. This is just impression I had of the fact that, yes, there are some organisms, staph, for instance, which affect humans and animals, but many don't. Many don't.
I guess
I don't know. I think that I'm coming down on the side that bacteria are pretty specific for the host.
The microbiome should be able to answer that, because as we characterize the human microbiome and we get a snapshot of the number of genera and species that are associated with us, and then we do the microbiome of, say, for instance, rats or mice, or pick your favorite animal that's even closely related to people like chimps, we may see very different things.
Yeah. That would be good. That's an unbiased approach to it, right?
Yeah.
Yeah, but then the distinguishing a species.
Yeah, right.
What's a species is going to complicate the answer.
We get into that argument.
Yeah.
An OTU, right?
An OTU.
Yep. All right. The last one is from David. Hi, guys. Sorry for such lag in response about the number of rats in New York. I'm uncertain. Heard a couple of million recently. However, I felt obligated to share an anecdote. Popular Canadian life scientist David Suzuki, in an interview with Bill Moyers a few years ago, stated, sharing an air of astonishment and awe, that planet Earth now has more humans than it has rats. This is noteworthy, of course, because rats are so much smaller, eat so opportunistically, and reproduce so fluorescently that you'd kind of think there'd be more of them than us overall. There must once have been. That was his impression anyhow, and I was willing to buy it. And it sort of left an impression of the massive quality of Earth's infestation with Homo sapiens at this point. Just think. So it's like you, Michael, when Bill Moyers or David Suzuki says something, people believe it, right? Now, the next part, he talks about the biofilm in his toilet. And I don't want to read it, but you guys, the listeners, if you want, you can go to the letters, and he's interested in analyzing it, but I don't want to read this. And then he says, one time I may tell you about the bioluminescent, presumably fungal growth observed in my compost heap while turning the thing in the dark of the less hot night a while back. And he gives us the weather report, and gratefully, Dave. P.S. Heaps of praise on your communication work. Well, thank you, Dave, and thank you, all of you, for those letters. If you would like to send a letter in and have it read and, get the expertise of real microbiologists, except me, well, I guess I could be a microbiologist, too.
Viruses are in there.
Oh, this is not becoming, Victor.
Elio, I'm in a microbiology department, so I must be a microbiologist. Right?
Absolutely.
Of course you are.
But also, the questions stimulate some nice discussion amongst us, and you can hear what we think. So do prime the pump. You send your email question to TWiM, TWIM, at twiv.tv. And this podcast, This Week in Microbiology, you can find it at iTunes. You can find it at microbeworld.org/TWiM. But more likely, you will use your favorite podcast listening program. There are tons of them out there for your iPhone, for your Android device, and they're free typically. And you can subscribe to podcasts, and they all get downloaded automatically. Very easy way to consume this content. I'd like to thank everyone for participating today. Michele Swanson is at the University of Michigan. Thank you, Michele.
My pleasure. Nice to chat with you all again.
And thank you for contacting the author of that lovely paper.
Yeah, we had a great conversation.
Cool. Elio Schaechter is at Small Things Considered. Thank you, Elio.
Sure was fun.
And Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM, and Chris Condian and Ray Ortega for their technical help. And Ray in particular, he edits every episode of TWiM, gets to cut out all the stuff that's wrong. Just kidding. But
this is actually a 6-hour podcast, and we distill it down.
So thanks a lot to Ray. The music you hear at the beginning and the end of TWiM is composed and performed by Ronald Jenkins. And if you like his music, go hear him. You can find it all at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.